All right, so let's go over the different types of cells in ground tissue. There's three types of cells. There's parenchyma cells, there's collenchyma cells, and sclerenchyma. So we're going to go over each one and their features. All right, so the parenchyma cells this means no secondary cell walls. All right, so that's what my shorthand means here. They do not have secondary cell walls, which means they only have primary cell walls, and therefore they can do mitosis. Now, this is an important feature because it means that if if a plant is cut, then these cells can fill in by doing mitosis from a neighboring cell and replacing the cells that got cut out or damaged. So this is why it's part of wound healing. They are alive at maturity. They are photosynthetic. So in a stem, if you look at a stem, remember that the epidermal cells, actually maybe I didn't say that, the epidermal cells don't really have very many chloroplasts. The guard cells do, but the actual epiderm cells, so these cells don't really have that many chloroplasts. In the case of the parenchyma cells, which would be just inside of that, they do have chloroplasts, and they are therefore photosynthetic. So if you see a stem and it's green, or a leaf and it's green, what's green about it is the parenchyma cells that are doing photosynthesis. And we'll talk more about that later, where the green comes from. All right. These also initiate adventitious structures like in cuttings. Like if you made a cutting, you stuck it in water, and the roots were to grow out of it. Those are called adventitious roots, and they are coming from the parenchyma part of the stem. So that is just inside the epiderm. Those cells that can do mitosis would be creating the new roots that would be growing down. They also are part of some specialized ones are called transfer cells. These are cells that have the cell membrane like inundated or like squished inside of the cell wall, meaning that water can pass between the cells easier in the middle lamella sort of continuum between cells. So it also makes a shorter distance across the cell for water to go into a cell and out the other side as well. So those are called transfer cells. They're sort of specialized. We probably will never talk about them again, but you should just have heard about them. Collenchyma are another type of ground tissue. These are cells that also don't have secondary cell walls, but they have uneven cell wall thickening. So if you look at it, it looks like they have secondary cell walls, but they don't. But because of this uneven thickening, they don't generally do mitosis. They are, however, alive at maturity, and they're very long cells longitudinally. So what happens is they span more than one of the neighboring cells in a long axis of the plant. And this makes them like supportive structures. So for example, they'll be on the outside of a vein of a plant to support that vein so it doesn't get broken. An example that you probably know are the strands that you find in celery. So you know when you bite into celery and those big long strands that come off, those celery strands are these collenchyma cells, all right? The last type is called sclerenchyma. Sclerenchyma cells do have secondary cell walls, and they are dead at maturity. You're going to see that's a theme. If they have secondary cell walls, they will be dead. Therefore, they don't do mitosis. And there are two types of them. There are fibers, which are very long. Again, they span more than one of the neighboring cells, but these are dead. So these are actually, more strictly speaking, supportive structures. And like in hemp, for example, the stuff that we make rope out of, those are the fibers that we're talking about. And sclerids are short ones. They're short and really hard. So when you eat a pear, that grit that you taste within the pear flesh is a sclerid. All right, so here's just an overhead of some of the cells. So parenchyma, see the chloroplast, photosynthetic. Collenchyma, this isn't a great picture of it. It's kind of dark, it's hard to see, but you will see some pictures of it in the lab. And then in sclerenchyma, they just look like little teeny short squatty cells within the other cells perhaps within the parenchyma cells of another type of tissue. All right?